in an experiment.
We don't know yet.
Why is light so far?
Like, it sounds so simple.
They had no idea.
But now the data speaks.
I find this not only refreshing, but at some level astounding.
Nature.
Welcome back to the Nature Podcast. This week, why leukemia risk is so much higher in children with Down syndrome.
And the latest from the Nature Briefing. I'm Emily Bates.
And I'm Benjamin Thompson. Down syndrome is a genetic condition often associated with a number of cognitive and physical health impacts. It's caused by having an extra copy of chromosome 21, meaning that 3 copies are present in each cell, known as trisomy. This week, Nature has a paper out that's been looking to understand something that's puzzled researchers, namely why children born with Down syndrome often have issues relating to their blood. For instance, many newborns have elevated levels of red blood cells, which often drop back to regular levels pretty quickly. But alongside this, there can be other issues too.
Maybe something that is less well known, but something that we were particularly interested in is that these children, they have 150-fold higher chance of developing leukemia compared to the children that do not have trisomy of chromosome 21.
This is Anna Sveich from the University of Copenhagen in Denmark, one of the authors of the new paper. The risk of developing a type of leukemia is hugely elevated for these children in the first 5 years of life, and Anna and her colleagues have been working to figure out why this might be, building on the work of other teams.
What we actually know now for decades is that all of these events that we are seeing early in childhood in children with Down syndrome are actually having their beginnings during fetal development, which will later on, in a certain sequence of events, lead to development of the leukemia.
Previous research established that cells at the centre of this sequence are hematopoietic stem cells, which have the ability to divide and develop into various types of blood cells, and established that certain genetic mutations played a key role. But Anna wanted to dig deeper into what's going on in cells that have an extra copy of chromosome 21, and how this might lead to leukemia and other blood-related issues. First, she and her colleagues took a snapshot of all the individual cells in fetal liver samples. Much of their work concentrated on the liver, as it's one of the main places in a developing fetus where blood cells are produced. This cellular census matched what others had found.
So what we were seeing is what other people also reported before, and that is that we are seeing increase in the number of stem cells, and we were seeing increase in the cells that will make red blood cells and platelets, and there is a decrease or impairment in immune cells like B cells. And they were using certain set of technologies to observe this. We are now using a different technology. So the first question is, can we actually see something that was described using other technologies? So if we can see what other people saw, that's kind of reassuring because it means that our analysis is kind of good enough to capture these changes. And it's a good starting point to then dig deeper into the mechanisms.
Levels of hematopoietic stem cells are known to increase in the liver of those with Down syndrome. But while these stem cells had the potential to become a number of different cell types, they were much more likely to follow the path that led them to becoming either red blood cells, also known as erythrocytes, or platelet-forming cells. To understand why, Anna and the team looked at the genomes of these cells to see what effect having an extra copy of chromosome 21 could be having. They showed that it was leading to changes in the way DNA is packaged. Now, DNA isn't just floating around inside a cell's nucleus as a simple double helix. Essentially, it's found wound round proteins, like string wound around a set of beads, which compacts and packages it into a structure called chromatin. This packing plays a key role in gene expression. Different sections of chromatin on different chromosomes are unwound at different times, allowing specific genes on the DNA to be turned on or off when required. And it turns out the presence of the extra chromosome was changing which DNA was available.
So basically, for a cell to be a stem cell, it has to have certain properties. And one of these properties is to have the ability to make many different cell types. But it can achieve that by having many different regions open, kind of having its options open, like which direction it's going to go. Whereas what we're seeing is a really kind of big restructuring of the chromatin accessibility in the stem cells, that they're narrowing down their options. So they're actually kind of preferentially opening regions which are going to regulate the gene expression of the erythroid lineage genes. And that is then causing higher expression later on of these erythroid genes and pushes these cells towards erythroid lineage.
These regions are where things called transcription factors can bind. You can think of transcription factors like on-off switches for genes. Changes to the chromatin opened up areas for them to attach, turning on genes related to the proliferation and differentiation of stem cells, which could help explain why high counts of erythrocytes, or red blood cells, are seen in newborns with Down syndrome. But what of the increased risk of leukemia? Well, by making use of previously collected data, the team identified that changing the chromatin structure altered the chances of where mutations to the DNA could occur.
We see that these mutations are preferentially happening in these areas in which these transcription factors are going to bind. So you can imagine if that is mutated, that can cause like transcription factor not to bind or to bind differently, and then can cause changes in gene expression. So at the stage when we are looking, none of these are causative of any phenotypes that we are seeing. But in the future, if some detrimental mutation happens in a right place, then it will cause pre-leukemia and leukemia.
The team didn't see this process happening, but their snapshot of the state of fetal cells gave a sense of what could happen further down the road if damaging mutations occurred, ultimately leading to genes being activated incorrectly. But what could be driving these mutations? It turns out that altering the chromatin structure is doing something else as well.
So one of the things that also became very clear that is happening in stem cells in the fetal liver was that these cells kind of had changes in a gene expression which suggested that there is an oxidative stress happening in these cells. And this comes from dysfunction of mitochondria. So these are what they call the powerhouses inside of the cell which provide energy for cell to function.
Oxidative stress is an imbalanced state where a cell has too many molecules called reactive oxygen species. These have a dual role. They can act as signalling molecules, but they can also act as molecular wrecking balls, smashing into structures like DNA and causing damage. These reactive oxygen species are thought to play a key role in introducing mutations into the genome of these cells with an extra copy of chromosome 21. And these different pieces, the oxygen species, Anna's finding about the opening up of certain areas of the genome, and that some areas are more prone to mutation, could all come together to help explain why leukemia risk is so elevated in children with Down syndrome.
The big strength of this paper is like providing the data on top of explaining many of the phenotypes for many research to come and then try to dissect even further.
This is Sébastien Melange, a molecular biologist at the Kids Research Institute Australia, who studies how leukemia develops in children. He's written a News and Views article about the new research. He was impressed by the work and the insights it provides into how blood disorders could begin to come about within fetal livers.
It was known that those disorders appeared extremely early, and it was known that it was due to the trisomy 21. But what was not known is how it works, right? And I think the strength of this work, the strong power of this work is to give clues about how those things happen. It's been done very deeply, right? They use single-cell sequencing to interrogate every single cell to know where those traits are coming from.
Sébastien says this research provides a strong foundation for future work to further unpick what causes the events to happen. But there's still much to learn. For instance, he'd like to get a sense of what's going on with the genome at an even earlier stage.
Personally, I would like to know what's happened before. So we see this snapshot of the open chromatin, but how does that happen, really? We don't know if it's like a big burst happening at one specific time, or if it's a slow process during the several weeks. We don't know. I think it's a very interesting question that it's going to be very hard to answer.
There are lots of other things to understand too. In this work, Anna identified a subpopulation of stem cells in the liver that seem to cycle a lot faster and have an even more pronounced bias towards ultimately becoming red blood cells. And then there's the fact that what was seen in liver stem cells was not seen in bone marrow cells, another place where blood cells are made. And other tissues can have different gene expression patterns. So what is it about the presence of the extra copy of chromosome 21 that's driving all this? Anna's got some ideas.
Clearly, because there is additional chromosome 21, there could be upregulation of the genes which are encoded by chromosome 21. And this is automatically going to change kind of regulatory networks. But you can also think that having additional chromosome for a cell is a burden because, a lot of things will change in the metabolism of a cell in terms of how much proteins there are and so on. And then you can also think physically, like how all of these chromosomes are packed in the nucleus. So you can think that having additional one can kind of cause some pressure like on a cell space-wise.
Finding out which combinations of these is the driver is going to be difficult. But it's important to remember that people are at the centre of this. Children with Down syndrome are at a vastly higher risk of leukemia, and both Anna and Sebastian hope this research will be a step in helping to understand why this is. Sebastian says that while leukemia treatments are available, ultimately not needing them would be a huge benefit to these children.
If we can decrease the higher risk of these children to have leukemia, that would actually be a long-term goal, right? And that's a very important step, this paper, to start providing some key features to find those indicators. So their treatment exists, so we save lives more than 50 years ago. But the price is the toxicity and the long-term secondary effect, right, of all of those toxic treatments. So whatever we can find to actually avoid to use life-threatening treatments is, a key in the future of research for leukemia specifically.
That was Sebastian Melange. You also heard from Anna Svejc. To read Sebastian's News and Views article and Anna's paper, look out for links in the show notes.
Coming up, how tiny crustaceans can smell their way home. Right now, though, it's time for the research highlights with Dan Fox.
Researchers have found a straightforward way to reduce beer consumption amongst English pubgoers. Take pints off the menu. At 568 millilitres, the Imperial pint is among the largest serving sizes of beer in the world. It's also the most popular serving size for beer in the United Kingdom, so much so that going for a pint has become shorthand for grabbing a drink. But alcohol overuse is a public health issue, spurring a search for ways to reduce how much people drink. Previous work had suggested that people drink less wine when serving sizes shrink. To check if this held true for beer, a team of researchers tracked alcohol consumption in 13 establishments that had replaced Imperial pint glasses with clearly marked 2/3 versions for 4 weeks. They found that during those weeks, patrons drank on average almost 10% less beer per day. And while wine sales rose, it was not enough to offset the drop in beer sales. The study suggests that shrinking serving sizes can reduce alcohol consumption. Drink in the full measure of that research in PLOS Medicine. In the tropical forests of Costa Rica, a small lizard has worked out a novel way to avoid predators: scuba diving. Semi-aquatic anolis lizards like the water anole can stay submerged for at least 20 minutes, using an air bubble on their snouts that allows them to breathe underwater. Now a researcher has tested whether these bubbles actually help them stay underwater for longer. By applying a substance to the lizard's skin that prevents bubble formation, they were able to see that without air bubbles, the lizards couldn't stay underwater as long as usual. They surfaced about 30%, around 67 seconds, sooner than lizards that could form bubbles. Next, the author plans to investigate whether the lizard's bubbles act as a physical gill, similar to those used by certain aquatic insects to extract oxygen from the surrounding water. Read that research in full in Biology Letters.
Finally on the show, it's time for the briefing chat, where we discuss a couple of articles that have been highlighted in the Nature briefing. Emily, why don't you go first this week? What have you got?
So I was reading a story in Science. It's all about these tiny crustaceans called mysids that use their sense of smell to find their way back to the caves that they live in. So every night, millions of mysids migrate into the open sea to feed. They leave at dusk and they return to their deep underwater caves before daylight to avoid predators. And it was known from previous research that they use light to navigate on their way out, but it wasn't known how they found their way back.
Right, and the ocean's a big place, especially if you're, a tiny crustacean?
1 millimeter, these little mysids, yeah.
So how did researchers go about figuring out that they were following their nose, so to speak?
So they took some of these mysids from 2 different underwater caves and they placed single animals in these Y-shaped containers. And sometimes one arm of the Y led to seawater taken from the mysid's home cave, the other to seawater collected from the other cave. Other times, neither arm led to water from the home cave, all kind of different configurations of that. But overall, the mysids sought out and preferred to spend time in water taken from their home cave. And then they also, in the second part of this study really, the researchers showed that water from each cave had its own distinct chemical signature or smell, which is what they think is guiding the mysids.
Right, so there must be something in there, a chemical X factor.
So yeah, we know salmon use chemical cues to find their way home in their migration, but now we're seeing it in these really tiny little crustaceans, which I just think is very, very interesting.
Presumably this chemical, whatever it is coming from the caves into the open water. Any idea what it is?
So it's not clear exactly what was giving each cave the sort of unique smell that was allowing these mysids to find their way, but the researchers hypothesise that it could be compounds released by sponges that live in the caves, and so the different amount of different kinds of sponges were giving each cave this very unique smell.
I mean, we say smell here. I think it's easy to sort of anthropomorphise or to, have a human-centric look at stuff, but is this actually kind of smell as we know it, Emily?
It is detecting chemical compounds and using that information going into the brain. We don't know how these animals perceive it, but it's as close to smell as we can imagine. One thing to think about around this is with marine pollution and things on the rise and sponge species, there's been a disappearance of some species from these caves. It could change the sort of chemical seascape of these underwater caves, and does that mean that in the future mysids won't be able to find their way home?
Right, which I suppose could have knock-on effects to the complex food web that exists under the water.
Absolutely.
Right, well, let's move on to my story this week, and it couldn't be more different. Now, it's a perennial favourite here on the Nature Podcast that we talk about ways to deflect asteroids that might crash into Earth and everything that goes alongside. And this is another one of those, and I read about it in Nature. It's based on a Nature physics paper, but it's looking at things in a slightly different way.
Interesting. Tell me more.
Well, so this work centres on whether a nuclear explosion could be used to deflect an asteroid. And that's the sort of thing that's been, thought about in Hollywood and in science for quite a long time, right? But in this case, it's looking at a different part of the explosion, the X-rays.
Oh, okay. So yes, I was aware of people talking about using a nuclear bomb to maybe, the impact to push an asteroid off course. But the X-rays...
Yeah, absolutely. So a lot of experiments have looked at the effects of how the momentum of a nuclear bomb shockwave that results from the expansion of gas could push against an asteroid, right? And kind of push it off course. But it turns out that nuclear blasts do create a huge amount of X-rays. And the researchers behind this work reckon that this actually might have a bigger effect on the trajectory of an asteroid. And so they set about testing it.
How do they test it? There aren't enough asteroids out there for us to go and send nuclear bombs up, I'm assuming.
Yes, admittedly, they did have to mock things up in a lab. And they used this really powerful bit of machinery called the Z machine, or perhaps the Z machine if you're from North America.
This is already sounding very Hollywood.
Well, this machine is amazing, right? It uses powerful magnetic fields to produce high temperatures and, of course, powerful X-rays. And one of the researchers involved said that about 80 trillion watts of electricity flowed through the machine in about 100 billionths of a second. These are numbers that are difficult to even comprehend, right? And this surge, what it does is it compresses argon gas into a plasma. And this plasma is this weird kind of state of matter, and it's millions of degrees in temperature, and it emits a bubble of X-rays. So that's how they made the X-rays. But the next step, of course, was to make some asteroids.
Right. What did they make the asteroids out of?
They made the asteroids out of quartz and silica, okay? So this reflects the different compositions of asteroids in the solar system, okay? And they made 2 of them, about the size of coffee beans. So just little tiny...
Little baby asteroids.
Little baby asteroids. And these asteroids were hung in a vacuum from thin pieces of foil in this machine. And when the X-ray bubble hit the foil, it acted as like a pair of scissors, I suppose, and cut through the foil. And the asteroids then put into free fall, and the researchers could see what the X-rays did.
Please tell me, what did they do?
Right, well, this experiment apparently only lasted 20 millionths of a second.
Okay, these numbers are incredible, okay.
But both the asteroids were accelerated to around 70 meters per second before being vaporized. But it seems that the cause of the acceleration, the researchers say, was these X-rays. And they were vaporizing the surface of the asteroids, causing, thrust as gas expanded away from the surfaces. So a little bit of a push.
Would this work on a larger scale? Because that sounds amazing, but does it scale?
Well, the researchers think that, yes, it could be scaled up to much larger asteroids, maybe 4 kilometers across, and sort of push them away from a potential collision course. But they do say that potentially it's most likely to have applications for big asteroids with a short warning time, the very worst ones, I suppose, because they think that maybe other approaches, like ramming a spacecraft into an asteroid, that's right, NASA's DART, they did manage to move an asteroid back in 2022, and we covered that on the podcast. But that approach might not have enough energy for these kind of short-term emergencies.
So is this something that we really think is going to be put into action? Are people excited about this idea, or is it Hollywood?
I mean, there's a lot of questions there. Will it be put into action? That's one of those ones I guess we won't know, and I also hope not. But looking at ways to deflect asteroids is kind of something that researchers are putting more and more time into, and certainly looking at how nuclear deflections might work, but also trying to figure out on Earth how you might test how effective these are, of course. But it seems that other researchers quoted in this news article are positive about the effectiveness of this as a technique. But the team behind it are looking to perform more tests, to refine it. And eventually they say, maybe, hypothetically, it could one day be tested in space, much like DART was, to see if it could nudge an asteroid.
I mean, that is amazingly cool. I hope we never have to use it.
Agreed.
Well, thank you, Ben. And listeners, for more on those stories and for where you can sign up to the Nature Briefing to get more like them, check out the show notes for some links.
And that's all for this week. As always, you can keep in touch with us on X. We're at Nature Podcast, or you can send an email to podcast@nature.com.
And if you fancy leaving us a review, you can do so wherever you get your podcasts. I'm Emily Bates.
And I'm Benjamin Thompson. Thanks for listening.